Hi everyone. Today I am going to start a new chapter, Cell, the unit of life. Under this, I will explain you about introduction part, historical events or scientist contributions, cell morphology and cell theory. G.N. Ramachandran, an outstanding figure in the field of protein structure, was the founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications and attention to solving the structure of collagen protein. Generally, unicellular organisms are capable of independent existence and performing the essential functions of life. Coming to the definition of cell, cell is defined as a unit of structure and function. Living things are made up of material units called cells. Study of structure of cell is called cytology. In addition to the structural and functional and hereditary characters of a cell is called cell biology. Therefore, sophisticated instruments and techniques are required to study the structure and function of cell and cell components. The living world is comprising organisms like microscopic bacteria to macroscopic multicellular plants and animals. Cells can be divided into two main types. They are primitive prokaryotic cells and evolved eukaryotic cells, which means cells are broadly classified into two types, prokaryotic cells and eukaryotic cells. Historical account are scientist contributions towards cell biology. Under this, the very first, the scientists F.Janssen and Z.Janssen invented first useful compound microscope. Robert Hooke discovered the cell in non-living tissue of cork slices, which means Quercus tree, and coined the term cellula or cell. Robert Hooke. These findings were described in his book, which is called Micrographia. Anthony van Leeuwenhoek observed living cells like bacteria, protozoa, RBC, etc. Desjardins named the living matter present in the cells, which is called sarcode. J. Purkinje. The scientist Purkinje coined the term protoplasm. Felix Desjardins termed the jelly-like material as sarcode. Sculge established similarity between sarcode and protoplasm of animal cells and plant cells. Later, this was improved and protoplasm theory was developed by Hooke. According to this theory, all living matter out of which animals and plants are formed is the protoplasm. Excluding cell wall, the plant cell is called protoplast. The term protoplast was given by the scientist Hanstein. General morphology of a cell. Cells that have membrane-bound nucleus are called eukaryotic cells, whereas cells that lack a membrane-bound nucleus are called prokaryotic cells. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm, which occupies the volume of cell. The cytoplasm is the main area of cellular activities in both plant and animal cells. Various membrane-bound cell organelles with their metabolic activities maintain the dynamic nature of the cell in eukaryotic cells. In prokaryotic cells, lack such membrane-bound cell organelles. Ribosomes are non-membrane bound organelles found in both prokaryotic and eukaryotic cells. Animal cells contain a membrane-bound organelle called centriole, which helps in cell division. Cells differs greatly in size, shape, and activities. Mycoplasmas are smallest cells measuring 0.3 m in length, while bacteria could be 3 to 5 micrometers. The largest isolated single cell is the egg of an ostrich. In multicellular organisms, human red blood cells are about 7 micrometers in diameter. Nerve cells are some of the longest cells. Cells also vary in their shapes like discoid, polygonal, columnar, cuboid, thread-like, or irregular shapes. Let's see the diagrammatic representation of various cells which are in various shapes. The very first one, RBC, red blood cells, which are in round and biconcave shape. Next one, white blood cells, WBC, which are in amoeboid shape. Columnar epithelial cells, which are long and narrow cells. Coming to nerve cell, which is long and branched. Coming to tracheid, it is long elongated cell. Mesophilic cells, which are round and oval shaped. Let's check these diagrams carefully. Red blood cells, round and biconcave. White blood cells, amoeboid. Columnar epithelial cells, long and narrow. Nerve cell, long and branched. Elongated cells are tracheids, and mesophilic cells are round and oval shape. Cell theory. M.J. Schleiden, a German botanist, and T. Schwann, a German zoologist, together formulated the cell theory or cell doctrine. Schwann proposed that the presence of cell wall is unique character of plant cell. According to cell theory, cell is a structural and functional unit of life. However, this theory did not explain how new cells were formed. Coming to Rudolf Virchow, first explained that cells divide and gives rise to new cells from pre-existing cells, which means omnis cellula e cellula. From his theory, cell lineage theory or cell inheritance theory. The scientist Rudolf Virchow modified the cell theory as follows. Cell is the structural unit of all living organisms. Cell is the functional unit of all organisms. Cell is the hereditary unit of all living organisms. And the important point he added to cell theory is omnis cellula e cellula, which means all new cells are formed from pre-existing cells. Up to here, cell biology introduction, historical events, cell morphology and cell theory was completed. In next video, I will explain you prokaryotic cell. Thank you very much.